Well, welcome to the Master Gardener module on basic nematology. Our learning objectives for this module are to define what plant parasitic nematodes are and how they damage plants. And we'll probably just call them PPN to make it a little bit easier. Recognize plant symptoms resulting from PPN damage. Know PPN management strategies for various crops. Describe how to solarize soil to reduce nematode populations, and be familiar with nematode sampling procedures and diagnostic capabilities provided by University of Florida Institute of Food and Agricultural Science. So we'll start with looking at the life cycle of nematodes, the plant damage and symptoms, the plant damage they cause, and the symptoms of nematode damage. Nematodes can be found from the depths of the ocean to mountaintops. A spadeful of soil can contain more than 1 million nematodes. PPN, plant parasitic nematodes, are unsegmented roundworms that differ from earthworms and other common worms. They are microscopic. They live in a film of water surrounding soil particles. They use spear-like mouth parts or stylets, seen right here, to feed on plants, and they inhibit root growth and plant performance. Where are they found? This graph represents the percent of nematode species, not individuals, but species, that live in different environments. In actuality, the majority of nematodes live on the ocean floor. You can see that on or in plants, about 10% of the species live on or in plants, 15% inside of animals. Good versus bad nematodes. Let's consider good ones first. Most nematodes feed on bacteria or fungi and are important in decomposition and nutrient cycling in nature. Some nematodes are predators, preying on microscopic animals, insects, or other nematodes. What's bad about them? Some of the nematodes that we think of as bad are animal parasites, such as heartworms, that cause diseases of humans, pets, and livestock. Here we're looking at the general life cycle of a PPN. It's relatively complex. You can take your time, but you see eggs are laid. They form larvae, many different stages of larvae. Finally, adults, which then can infect more roots. Note, nematode reproduction increases with increasing soil temperatures and decreases as the soils cool. How do they feed? They use stylets, we see here. Resembles a hypodermic needle, syringe. It's inserted into a plant cell. Digestive juices injected into a cell. So these are enzymes. These are enzymes that are going to break down proteins, break down carbohydrates. And then once those proteins and carbohydrates are hydrolyzed, the plant juices are ingested through the stylet, sucking them up like through a straw. Where do they feed? Most feed on roots, plant roots. There is an exception, Aphelinocoides, which feeds on foliage. Foliar nematodes are economically important in some nursery crops, such as chrysanthemums, ferns, anthuriums, and azaleas. They can be ectoparasitic or endoparasitic. Ectoparasitic means they feed outside of the root, as you can see here, with the stylet going into the root tip. Endoparasitic means they feed inside the root, and we see one inside a root here. In Florida, the important ones, ectoparasites we see are the sting, stubby root, spiral, and ring nematodes. There are also migratory endoparasites. So these are inside the root, but they move. These are lesion, lance, burrowing, and foliar nematodes. And there are other endoparasites which are sedentary. Once they get into the root, they do not move. And this will be root knot and citrus. They damage plants by reducing the plant's abilities to absorb water and nutrients. This damage exposes plant roots to infections by diseases. Here we see the difference between healthy, normal roots and infected roots. And there's another example of an infected root, the stubbiness of it. So let's look at some symptoms of decline. Above ground, you can see yellowing, nutrient deficiencies, stunting and slow decline, premature wilting and slow recovery, thinning, or even death. But note that these symptoms are similar to those caused by drought, other diseases, or just general stress. Below symptoms vary with the type of nematode and plant. General root symptoms include stunted and shortened roots, deformed roots, dark or rotted roots, lesions and galls, or reduced root proliferation. Here we see healthy roots, damaged roots. They can cause splitting in carrots, and here we see deformed roots. The rate of decline depends upon the species of nematode, how many there are, the population density, the susceptibility of a plant species or variety, the soil temperature and texture, and the presence of other stresses. So now let's go to part 1 of management of PPN in the vegetable garden. So you see a problem, is it nematodes or something else? Some symptoms may be obvious. Considering root knot nematodes, there are 3 common root knot species in Florida, and they are recognized by galls on fibrous roots. The galls can be very small, but they may enlarge, they may merge together and enlarge. Susceptible plants include those in the Solanaceae, tomatoes, potatoes, okra, the legumes like beans, cucurbits, squash, cucumbers, and carrots. And here you can see some of these nodules or galls. Here we're looking at an egg mass and an enlarged adult female. There are also some not so obvious symptoms. In tap roots, galls may not be as visible and the roots can get knobby, as we see here. In onions, the galls are barely noticeable, but the bulbs can be discolored and smaller, so healthy, infected. And in potatoes, the appearance is marred, and a cross-section could reveal speckling or feeding sites. See all the stained areas here, and the marred appearance. It's not smooth. Root knot nematode galls may be confused with beneficial nitrogen-fixing rhizobium nodules on legumes. Rhizobium nodules are modified root hairs and therefore can be pulled easily from the main root. Root knot galls are formed from root tissue, so they cannot be pulled off. So rhizobium nodules, much different appearance than root knot galls. This slide's a bit difficult to read, but you can see it in your handbook. It's Management of Vegetable Garden, your Master Gardener handbook. It's the Management in the Vegetable Garden, Table 1. We can see, though, that root knot and stings affect almost every one of these vegetable plants. Stubby roots get a lot of them, but then the other different types of nematodes are very specific, not nearly as generalist as the root knot and the sting. There is a bionematicide called Melocon, WG, which contains a fungus that parasitizes nematode eggs. Results from University of Florida trials indicate that Melocon can suppress but not eliminate root knot and sting nematodes on vegetables and shallow-rooted perennials. If you're going to purchase Melocon, have it delivered directly to you from the manufacturer. Keep it frozen until it is used. To apply it, you mix it with water and then either drench the planting bed with it or spray it onto the planting bed and irrigate. Melocon works best when it is administered in multiple applications, starting before transplanting and continuing through the growing season. It is generally not effective when used to treat established plants after they have been damaged by nematodes. So this is one management option for vegetable garden. There are no chemical nematicides. There are some bionematicides. You can also try using sanitation methods, so don't introduce contaminated plants or soil. Remove plants and roots after harvest and keep your garden weed free. You can solarize the soil, which we'll learn more about. We'll also learn more about using cover crops and organic amendments and crop rotation and resistant varieties. Now, I did look up Melocon, and it's about $350 for 12 ounces, I think it was. It came out to be right around $10 an ounce, a little bit more than $10 an ounce, and you use 1 ounce per acre. So let's consider sanitation and prevention. Nematodes move very slowly on their own. They're moved about by soil, by water, by plants and or tools. So power wash or disinfect your tools with a bleach solution. Remove nematode-infected roots after harvest and do not compost them. Nematodes spread on tools, shoes, wheelbarrows, vehicle tires, people, pets, improperly or partially decomposed compost, infected plants, imported soil, and water. So they get around with a lot of help. So let's look at soil solarization. What we're going to do is work and moisten a patch of soil. This activates the pathogens and weeds and also increases heat conductivity. Then we're going to cover that patch of soil with clear plastic or thermal infrared retentive film. We're going to seal the plastic on the soil to prevent heat loss. And we want the soil temperatures to reach 95 to 140 degrees Fahrenheit, with 135 being ideal, and maintain those temperatures to a depth of 4 to 12 inches. The plastic is left in place from at least 4 to 6 weeks. The longer, the better. This is a simple illustration of plastic solarization over here. And this is just the thermometer showing the temperature of that soil. This practice works best in summer, of course. Depends upon heat. It does not work well in shaded areas because they're not going to get as hot. It affects everything, all living organisms in the upper 6 to 8 inches of the soil. This is weeds, disease pathogens, insects, nematodes. And it also affects beneficial organisms negatively, but they can recolonize quickly. It has no significant impact on organic matter decomposition. And after solarization, you don't want to mix or till the soil again. There are numerous, if you do a search, there are numerous guides for solarization available. Root knot nematode eggs and juveniles are very susceptible to heat over short periods of time. Meloidogyne incognita, 390 hours at 100 degrees Fahrenheit. So it takes 390 hours at 100 degrees Fahrenheit to kill the eggs and juveniles. But at 108 degrees, it only takes 15 hours. For a different species, the juveniles were killed instantly at 122, and the eggs died after 2 to 3 weeks at an average temperature of 109. Most PPN species recolonize soil to damaging numbers by the end of the crop cycle following solarization. So you solarize, you plant your crop, and by the time you're done harvesting that crop, the nematodes are going to be back. So one thing you can do is follow solarization by a cowpea cover crop, and this suppresses the root knot nematode population to a level equivalent to a methyl bromide treatment. And methyl bromide is the gold standard, although it's damaging to the environment. It's a gas treatment of the soil, kills everything in the soil. And there's been a lot of work to try to find other methods so that we don't have to use methyl bromide, especially commercially. Let's look at the properties of organic amendments. If it's added as compost, manure, or green manure, etc., it enhances soil microbes and beneficial nematodes. Some decomposing plants release chemicals toxic to nematodes, such as the brassicas, the cabbages, the Brussels sprouts. Those are all brassicas. But so even if the nematodes are unaffected or not affected, the organic amendments benefit the soil and plant health. Let's look at cover crops for a second. Cover crops are grown between vegetable growing seasons. So you harvest, you plant a cover crop, and you leave it in place until the next planting. They improve soil fertility and structure. They decrease erosion. They suppress weeds, insects, nematodes, and disease. They may be incorporated as green manure, so you till them into the soil. You enhance many beneficial organisms, and legume cover crops actually also supply nitrogen to subsequent crops because of the nitrogen-fixing bacteria associated with their roots. And here we're looking at a cover crop of cowpea. Here are some different nematocidal cover crops. Cowpea, the one we just saw, and I guess it depends on if it's going to get root knot nematodes. It doesn't affect sting nematodes. Sun hemp, which is a crotalaria species. Sorghum. Marigolds. And marigolds depend. Some marigold varieties are resistant, others are susceptible. Velvet bean, rye, and I don't know if that's Bahia or Bahia. Paspalum notatum. That'd be one way to find out. Now these are all references to EDIS documents, and EDIS, E-D-I-S, you can find them online. What it stands for is Everyday Information Source, or more simply known as EDIS. A few more nematocidal cover crops: indigo, Pangola grass, and a crotalaria. Here we're seeing some images of marigolds. Here it's just planted, it's a solid cover crop. And here they're planting it, it's interplanted with the crop. And here's a specific EDIS publication for that. And what they're showing you here is this is not the way to do it. If you're going to do a cover crop, do it this way. You can use resistant varieties. There are vegetable varieties that are resistant or tolerant. And look for the VFN designation on the seed pack or the tag. And this is Verticillium wilt, Fusarium, and nematode. And use fruits or ornamentals, or you can use fruits or ornamentals grafted on nematode-resistant rootstock. There are some nematode root knot resistant tomatoes listed here, but this only refers to 1 or 2 species of root knot nematodes. They could still be damaged by other species of root knot nematodes or other types of nematodes. Here we're looking infested cucumber roots. See how deformed they are? So we destroy nematode-infected roots and root fragments, which serve as reservoirs for nematodes. Tilling non-infested roots into the soil will improve soil health. So here it just looks like it's just been cleaned of all the vegetative matter, all the roots. If all else fails, you can try container gardening. But the container should not be in contact with nematode-infested soil. So if you're going to use containers, do it this way. You can see here they're on a surface. They're not sitting on the soil. I use paver blocks under the containers to keep them off of the soil. Use pest-free growing media, disinfected tools. Make sure you use disinfectant tools. And if you're doing container gardening and soil gardening, keep the tools separate. If possible, isolate the containers with screen cages, etc., because wild animals can come in and dig up the soil. So your first activity, dealing with crop rotation and resistant varieties. Many types of PPN damage vegetables, particularly root knot nematodes. One method of reducing damage is to rotate crops in the garden and to plant more nematode-resistant vegetable varieties. So now you can refer to Activity 1 worksheet in your Master Gardener Handbook. Well, now that you've completed Activity 1, we can move on to Nematodes Part 2, the management of PPN in turf grass. Here we're looking at symptoms of decline in turf grass, the above ground symptoms. You'll see irregularly shaped patches, like what we see here. There'll be yellowing, wilting, and browning. Grass dies or thins, and weeds thrive. And you'll get depths of whole areas, as seen here. But be careful, because this is very similar to drought damage, insect damage, disease, or just general stress. Below ground symptoms. Roots are abnormally short and stubby, darkened or rotten. Roots appear cropped off just below the soil surface, what we see here. Usually you don't see root galls or knots, though, on turf grass. Note the irregular areas of declining turf. It's not like a circle where chinch bugs would be getting it or sod webworms. And the damage can become more severe if there's drought or if there are underwater restrictions, which of course we are. They can be introduced from sod coming from a sod farm. So they can be introduced into landscapes in new sod. This is from EDIS publication on nematode management in residential lawns. We're looking at various types of grasses, whether they're susceptible, what nematodes, and some notes. Note that Bahia is the most tolerant turf grass. St. Augustine is commonly damaged, but Floratam is tolerant to sting nematodes. And we have Zoysia. There's no difference among cultivars. Bermuda is commonly damaged. There are some more tolerant. Centipede commonly damaged, very susceptible to ring nematodes. Seashore paspalum, I'm not sure which one that is, and it depends. Tolerant to most, very susceptible to spiral, can be damaged by lance. Of course, St. Augustine is probably the most common residential turf grass. Certain weeds tolerate nematodes and can proliferate as the grass thins. So weeds can serve as reservoirs because they're tolerant as the grass declines. And some common weeds, we all know nutsedge, spurge, and Florida pusley, or Florida snow, as we know it. University of Florida research has shown that overseeding can increase nematode numbers on Bermuda grass by providing an alternate food source to the nematodes during winter when nematode activity subsides. Therefore, it is best to avoid overseeding when possible from a nematode management standpoint. So if you're going to manage nematodes in turfgrass, choose a tolerant grass, and this is part of the right plant in the right place, the Florida-friendly mantra, right turf, right place, whether it's sun or shade. Don't overseed in winter if nematodes are a problem. Use organic soil amendments. These can be manures or sludge. Increase soil water and nutrient holding. They increase soil water and nutrient holding capacity. They may increase plant tolerance to PPNs and may stimulate natural enemies of PPNs. And of course, proper maintenance of all your garden equipment, such as mowing, and lawn, such as mowing, irrigation, fertilization, reduces stress and promotes vigor, root vigor. There are some, as of 2017, there are 3 products labeled for residential turf grass. And I looked up these also, and they're very expensive. We're talking $300 to $400 for, a quart or so of the concentrate. I'm not sure exactly how much you would need. I didn't look at the usage requirements for them, but they're root knot, sting, and lance, root knot and sting, root knot, sting, lance. This is a protectant. These are contact or contact and systemic activity. They're long-lived, but this is not. Well, by long-lived, they mean 8 months. This only has 4 months of activity. There is caution for using them. They're not restricted use pesticides. So let's move on to part 3, management of PPN in ornamental plants. These landscape plants are among the most commonly damaged by root knot nematodes in Florida. These plants should be avoided where root knot nematodes are known to occur. And that's your butterfly bush, hibiscus. Roses and gardenias are okay because they can be grafted onto resistant rootstocks. But also boxwood, Pittosporum montana, Japanese holly, and ixora. So if you know you have root knot nematodes, be careful. There are some landscape plants resistant to 3 or more species of root knots, and these are results from UF research. That's Walters viburnum, sweet pepper bush, firespike, gold dust croton, and oak leaf hydrangea. There are some bedding plants that are very susceptible to root knot nematodes, such as coleus, pansy, snapdragons, petunias, nasturtiums, sunflowers, zinnias, and others. And here's Anita's document, a couple documents you could look at for more information. So how do we manage nematodes in ornamental plants? There are no chemical nematicides. There is the bio-nematicide, Melicon. So don't plant susceptible or infested plants where you know you have nematodes. So it's right plants for the right place, Florida friendly. Rotate bedding plants, alternate them with resistant or tolerant species and varieties. Solarize your annual plant bed. Add organic amendments. Remove infested roots and soil, or grow in containers. I like to grow in containers because I don't like to weed. But you don't have a large area of pollinator-friendly plants then either, in color. So now part 4, how do we sample for plant pathogenic nematodes? In the vegetable garden, you sample at the end of the crop. Both soil and roots are needed. You collect samples from around the drip line of each plant, sample down to a depth of 8 to 10 inches, but discard the first inch of soil. So it sounds like a core would be the best way to do it. Make sure roots come from the plant being sampled. Place soil and roots in the same plastic bag. Need a minimum of 2 cups of soil and 1 to 2 cups of roots. Follow care and shipping as instructed by the lab. And remember, the accuracy of the diagnosis depends on the quality of the sample. And there is a University of Florida lab that you can send your samples to, and along with a check. And you complete a form, download a form, fill it out, print it, fill it out, send it in with it. And they can look and see if you have nematodes, which kind you have, and the numbers. We'll get into that here shortly. If you're sampling in turf grass with existing damage, so you can see there's damage. So you're going to collect the samples instructed by lab, and they provide instructions online. Don't sample inside these dead areas. Sample because there's no nematodes there. There's nothing for them to feed on. Collect samples from the declining areas, the green edges. That's where they'll be feeding. Use a shovel, trowel, or core sampler. Collect from about 20 spots. Combine the soil in a bucket, gently mix, and remove 1 pint for analysis. Place the sample in a plastic bag sealed securely, and if it's a Ziploc bag, tape the zipper, and label it with permanent marker. Store it in a cool place, preferably cooler or fridge until shipping, and it's best to ship as soon as possible and preferably overnight. And again, the accuracy of the diagnosis depends on the quality of the sample. So if the turf grass has no symptoms, or if it's before you plant or lay sod, this is the method used. Again, collect the samples instructed by the lab, and the instruction is to sample in a zigzag pattern we see here. Use your shovel, trowel, or core sampler, 20 spots. Combine it in a bucket, gently mix, remove 1 pint for analysis. Place it in your plastic bag, seal it securely, label it with a permanent marker. Store in a cool place, preferably cooler or fridge until shipping, and ship it as soon as possible, preferably overnight. And the whole thing here is you want to keep these nematodes alive. They can't tell you if you have them or what they are if they're dead. And again, the accuracy of the diagnosis depends on the quality of the sample. I actually did this in my yard last year, and it was very enlightening. I found out that I did have lance nematodes that could become damaging under stress conditions. Here we're looking at nematode counts from 4-foot squares over a larger area to show you how uneven the population densities are and why it's critical to sample correctly. You can see here in this 4x4 area we had 470, a count of 470 nematodes. But here, just 4 feet away, we only had 4. And another 4 feet away we had 439. That's why you need to take those 20 core samples. Oh, this was why you need so many core samples, recommending 20. And this is turf, so you're sampling 3 to 4 inches deep. And again, accuracy depends on quality. When sampling for nematodes, it's important to sample where most of the nematodes are. Failure to do so will dilute the sample and make an inaccurate diagnosis. Pre-plant samples are normally taken 8 to 10 inches deep. Post-plant samples are normally taken where the majority of the roots are. So for an annual crop, that's 6 to 8 inches. Trees and shrubs, that's 8 to 10 inches. And for turf, that's 3 to 4 inches. You also want to sample at the right time. The nematode populations fluctuate throughout the year. They may be undetectable during winter and early spring, but increase to very high population levels by early fall. And here's the sample processing at the University of Florida Nematode Assay Lab. It looks like they wash it, they centrifuge it, They resuspend with a sugar suspension, wash it through a fine sieve, remove the sugar with fresh water, then they count under a stereo microscope. For roots, that's for soil. For roots and corms, etc., they filter it, goes through a screen, into a mist chamber for 72 hours, then count under a stereo microscope. So here's the things you don't want to do. This is 10 ways to destroy a nematode sample. If you dry it before you bag it, you're going to destroy it. If you put it in a large box so it can rattle around during shipping, you're going to destroy it. I guess they're going to get crushed some. If you take multiple samples and don't label the bags and let the lab figure it out, well, they're not clairvoyant. If you put it in a Ziploc bag but don't tape the zipper, you're just hoping that the bag won't open up in transit. Don't send grass clippings instead of soil. Don't collect less than you need a pint. Don't sample in areas that the grass is dead. Don't keep the sample on your dashboard to bake in the sun. It's just going to kill the nematodes. Make sure you send out a filled out form and payment with the sample. And when I did it last year, it was $35. Send the sample. Don't send that out on a Friday or just before a holiday, so it's going to sit there for 3 or 4 days before anybody gets a chance to process it. And here's an example of the results. You'll get the number of nematodes per 100 cc of soil, or the number of nematodes in 10 grams of roots, and they'll tell you the risk level measured against known thresholds for a specific host. And you can see it looks like the sting nematodes are the, you're looking at St. Augustine here, it's the sting nematodes that are the most detrimental. It doesn't take very many of them to have a high risk threshold, to get over a high risk threshold. Lance, stubby root, lance spiral seems to be the least harmful. And stubby roots are pretty bad too, about the same as lance. So now you can do activity 2. Since there are many types of plant parasitic nematodes in Florida soils, and symptoms can resemble disease or improper cultural practices or harsh environmental conditions, its laboratory analysis is necessary to determine if nematodes are causing a problem. So with that said, refer to activity 2 in your Master Gardener Handbook. And with that, we'd like to acknowledge the people who put this PowerPoint presentation together, this module, Dr. Billy Crow, and Roy Levin. Also, again, Sydney Park Brown. Lab activities were designed by Beth Bowles, and this was reviewed by Lisa Hickey and Julie McConnell. And with that, we'll close and look forward to seeing you again for Module 3.